Welcome to Neuro Insights. I'm Rittika, and today we are exploring gene therapy's cutting edge application in Huntington's disease, a monogenic disorder offering unique opportunities for precise interventions. Our guest, Kanak Chaudhary, holds a Master of Science in Biotech and offers insight from genetic engineering purposes and perspective. Kanak, thanks for being here. And to open the floor, I would like to start with a question, which is could you firstly describe, let's start from the very basics, the CAG repeat expansion in Huntington's, uh, or rather the HTT gene, and how it leads to mutant Huntington's toxic gain of function in striatal neurons? So there are many heavy terms, so it would be good if you could break it down.
Okay. Thank you so much for having me again. Huntington's arises from an abnormal expansion of these CAG repeats in the HTT gene, which translates into an elongated polyglutamine track in Huntington protein. This mutant form misfolds, aggregates, and disrupt the key cellular functions like mitochondrial activity, axonal transport, and proteostasis. And if we talk about the striatal neurons that are especially hyperbolized in because of their high energy demands and reliance on tightly regulated protein networks, making toxic gain of function the driving force behind neurodegeneration is one of the key.
Right. So let's talk about CRISPR applications. So CRISPR-Cas9 variants like base editors target repeat expansions. What nuances in off-target effects and delivery via AAV vectors are critical for Huntington's? And how do prime editing advancements address somatic instability in repeat regions?
So if we talk about the CRISPR-Cas9 variants uh, that have been adapted to target repeat expansions directly, the challenge is precision. The off-target cleavage in neuronal genomes could be devastating. The AAV vectors, while effective, raise some concerns about immunogenicity and limited cargo capacity. Base editors and prime editors offer more nuanced approaches, while the prime editing in particular is attractive for Huntington's because it can correct or contract repeat regions while reducing the risk of double-strand breaks, and it directly addresses somatic instability, which is a hallmark of disease progression.
Right. So I think it's a very promising avenue. So I think the next flow would be towards RNA-based therapies. So antisense oligonucleotides, also known as ASOs, and siRNA or small interfering RNAs, silent mutant HTT gene. So how do allele-specific design distinguish expanded from normal alleles, and what intraventricular infusion trials reveal about their penetration and long-term efficacy in primate models?
So the ASOs, the antisense oligonucleotides and siRNAs work by silencing mutant HTT transcripts before the translation process. And allele-specific designs exploit single nucleotide polymorphisms linked to expanded alleles, allowing them to selectively knock down the mutant while sparing the wild type allele, which has critical cellular functions ahead. And intraventricular infusion trials in primates specifically have shown that ASOs can penetrate the striatum and cortex while measurable reductions in Huntington. However, long-term efficacy still hinges on repeat dosing and ensuring durable effects without off-target silencing.
Right. So that is a very vital progress. And beyond editing, I think gene replacement has a newer prospect. So viral vectors for delivering truncated HTT gene or neuroprotective genes like BDNF are in development. So what challenges in immune responses and vector tropism arise, do you think? And how do nanoparticle conjugates enhance targeted delivery to the basal ganglia?
Another approach is to deliver truncated versions of HTT that retain essential functions or to introduce neuroprotective genes like you said BDNF. And AAV based delivery shows that the potential is there but comes with hurdles like immune responses against the capsid proteins, the tropism challenge, and since not all vectors effectively and efficiently target the basal ganglia, it is one of the problems also. And the nanoparticle conjugates are an emerging solution of it, like offering tunable surface chemistry for crossing the blood-brain barrier, and and it prefers the delivery to striatal neurons also, which is very helpful for the process, and they may complement the viral systems or serve as safer standalone carriers.
That was actually a very thoughtful answer. I have another question, which is regarding patient-derived iPSCs and cerebral organoid models of Huntington's. How do these platforms test gene therapies' impact on proteostasis and synaptic function? And what epigenetic markers like DNA methylation patterns serve as surrogates for therapeutic outcomes?
So, as you said, the patient-derived iPSCs and cerebral organoids have become more powerful test beds nowadays. And AAV based delivery shows that the potential is there, but comes with hurdles like immune responses against the capsid proteins, the tropism challenge, and since not all vectors effectively and efficiently target the basal ganglia, it is one of the problems also. And the nanoparticle conjugates are an emerging solution of it, like offering tunable surface chemistry for crossing the blood-brain barrier, and and it prefers the delivery to striatal neurons also, which is very helpful for the process, and they may complement the viral systems or serve as safer standalone carriers.
That was actually a very thoughtful answer. I have another question, which is regarding patient-derived iPSCs and cerebral organoid models of Huntington's. How do these platforms test gene therapies' impact on proteostasis and synaptic function? And what epigenetic markers like DNA methylation patterns serve as surrogates for therapeutic outcomes?
So, as you said, the patient-derived iPSCs and cerebral organoids have become more powerful test beds nowadays. They recapitulate the mutant Huntington aggregation and allow us to track how gene therapies restore the proteostasis and improve the synaptic signaling. Importantly, these systems also let us monitor the epigenetic changes, like likewise the DNA methylation and histone modifications, which can shift with treatment. Certain methylation patterns are emerging as biomarkers, serving serving as measurable surrogates to indicate whether a therapy is effectively modulating the disease pathways before the clinical endpoints are apparent.
Right. So innovative and I want to move on to moreover combination approaches. So, integrating these therapies with small molecule targeting autophagy. On this note, what synergistic effects emerge in clearing aggregates, and how do high throughput like CRISPR screening identify modifiers or personalized regimens in polyglutamine disorders?
So the gene therapy alone may not fully resolve the aggregation part, which is why combining it with the small molecules that enhance the autophagy is exciting. The synergy lies in reducing the mutant protein production while boosting clearance pathways, leading to more efficient removal of aggregates. Also, the high throughput CRISPR screens have identified modifiers, genes that, when perturbed, enhance the autophagy and reduce toxicity, which is there. This paves the way for designing personalized regimes where the gene editing is tailored alongside small molecule modulators for polyglutamine disorders.
Great. So it's a very exciting horizon indeed. Now, going into ethics, which is also as important and one must keep it in mind, ethical and regulatory hurdles in gene therapy trials for Huntington's are significant. How might AI optimized vector design accelerate safe translations, and what global equity issues in access to these therapies demand attention in biotech?
So it is a very good aspect to move on. The ethical and regulatory hurdles remain central to the Huntington's gene therapy. Like AI-driven approaches such as vector optimization are helping us design safer and more efficient delivery systems with reduced immunogenicity. Yet beyond the science, we must address the global equity. These therapies are likely to be expensive and without frameworks for accessibility, they risk deepening healthcare disparities. Ensuring equitable access and transparent regulation will be as important as the scientific breakthroughs themselves in making gene therapy a true solution for Huntington's patients worldwide.
Kanak, you have really advanced our view on gene therapy for Huntington's, and I really, really appreciate it. And to the listeners, stay tuned for the next episode.